Howdy! I'm going to give you guys some clues on what to do for your diffusion lab. So there's going to be two different setups. This is setup A right here, and you're going to get this dialysis tubing, and the most challenging part is just opening it. But you have to somehow get it open, tie off one side, and you're going to put some glucose solution in there, and then tie off the other side. And then you have to actually weigh it on one of the scales. Then you will put it in here, and you're going to let that sit for 20 minutes, and then you'll take another measurement, and you're going to be writing down those measurements on this chart right here. So write down the initial mass, and then after 20 minutes, and then we'll actually wait again after 24 hours and see what its mass is. And since this is a glucose or sugar solution, we're going to find out where the water went, but also where the glucose went. So you can actually take a glucose test strip and dip it into the water after 24 hours. Just stick it in there and then let it sit for a minute and see if there's any sugar in the water, to see if any left the bag. Then the second setup is starch. So you're going to put the starch solution in this bag, and you can see it already started settling a little. So once again, take the mass, write it on the chart, pop it in there, but there is one extra step. You actually have to put some iodine. So just put enough drops until it's all yellow. And if you remember the reaction that happens with iodine, it turns a bluish purple in the presence of starch. So if any of that iodine goes into that bag, it should turn a bluish purple color. And in fact, I could probably wait for a minute and it would do that. Here's a few extra tips for your worksheet on your lab. As you know, the membrane is a porous membrane. It's flexible and it is selective, controlling what goes in and out. And if you look at the different size molecules we're working with in this lab, I kind of put them in size order from smallest to largest. So water molecules are probably the smallest, then iodine, then sugar, then starch. In fact, you might discover that starch molecules are so big they can't even leave the membrane to get out into the rest of the solution. And since these cells are not actually alive, obviously this would be passive transport. It's just going with the flow. If it were to be active transport, it would require energy to go against the flow if you're trying to get something to go against the concentration gradient. But since these are not real cells, there's no energy to be had. And then the last concept is, if you have two solutions, one is a weaker solution, one's a little bit stronger, we know water always wants to flow to the greater concentration, so most of the water will flow towards the 20%, and then a little bit might flow towards the 10%. But eventually, if there's some going on both sides, if this was like a sugar solution, for example, some of the sugar molecules might eventually go across the membrane as well and balance this out till both sides were about 15%. So quickly here, finishing this up, you can see some of the iodine has already gone through the bag and is starting to turn the starch purple. So good luck with this lab.